Welcome to this, the 22nd Teagasc Research Insights webinar. My name is David Kenny, and I'm head of the Teagasc Animal and Bioscience Research Department. I will be chairing this morning's proceedings. This series of webinars focus on addressing the challenges and opportunities in the agri-food sector and showcase the latest Teagasc research. In this morning's webinar, we will highlight the work being undertaken by Teagasc scientists in conjunction with their national and international collaborators on a very important topical issue for the Irish agri-food sector, improving the environmental sustainability of ruminant livestock-based production systems. Specifically, our speakers this morning will outline their research work on strategies to reduce emissions of the potent greenhouse gas methane from Irish beef, dairy and sheep farms, while at the same time maintaining high animal performance, health and welfare, and of course, the production of quality food products. So then moving on to our panel this morning. First up is Professor Sinead Waters, who is a principal research scientist at the Teagasc Animal and Grassland Research and Innovation Centre, Grange, County Meath. Sinead will give a brief overview of the importance of reducing methane emissions from the Irish livestock sector, and in particular, will speak about some of the ongoing Department of Agriculture, Food and the Marine research funded projects which she leads. Following on from Sinead, Dr. Ben Lahart from Teagasc Moorpark will speak about his postdoctoral research on measurement of methane emissions from grazing dairy cows. And finally, Professor Donagh Berry, Senior Livestock Geneticist at Teagasc Moorpark and Director of the Science Foundation Ireland funded Vista Milk Research Centre, will outline the role of animal genetics and genomic technology in mitigating greenhouse gas emissions from cattle and sheep. Each presentation will be 15 minutes in duration. We're looking forward to a lively and interactive session, so please type your questions for our speakers in the Q&A function below. We'll attempt to address as many of your questions as possible during the discussion section, which will follow at the end of our last presentation. I would ask you to please also include the name of the speaker to which you would like to address your question to. The webinar is being recorded and will be available for viewing on www.teagasc.ie. So, without further delay, I'd like to ask the first of our speakers this morning, Professor Sinead Waters, to present her work. Over to you, Sinead.
Thank you, David. And good morning, everybody. So this morning, I'm going to talk about methane emissions, as David said, and particularly the development of novel technologies to reduce methane emissions from Irish agricultural systems. So as an overview, I'm going to cover methane emissions, how they're produced, and how we're going to potentially mitigate them. So I'm going to talk about strategies to reduce methane emissions from agriculture. And today I'm going to particularly focus on feed additives. And as David mentioned, the Methane project, which is funded by the Department of Agriculture, Food and the Marines. I'm also going to touch on another strategy that we're hoping to use, which is early life intervention and supplementation in early life as another approach to reduce methane emissions from agriculture. So what are methane emissions? Well, first of all, methane is a potent greenhouse gas, and agriculture is responsible for nearly a third of Ireland's greenhouse gas emissions. And these mainly include nitrous oxide, generally produced from fertilizers and manure, methane, which is produced mainly by rumen fermentation or the digestion of feed, and I'll talk about that later, and also carbon dioxide. So sources of methane from Irish agriculture include, as I mentioned, enteric fermentation, and this is the main majority of the methane produced in Ireland from livestock. It accounts for 56.2%. So enteric fermentation refers to feed digestion in the rumen, which is really the largest stomach of the animal. And this feed digestion is an essential process of converting feed or grass into high quality meat or milk for human consumption. But that comes at a cost, as methane is a byproduct of that process. Methane is also produced by stored slurries and manures on farms. We're under huge pressure to reduce methane emissions from agriculture, and this will be key to meeting our targets on climate change. So how is methane produced? Methane is produced by a process called methanogenesis. And as I said here, it occurs here in the rumen of the animal, or the largest stomach of the gastrointestinal tract in the animal, in ruminants, which are cattle and sheep. And this occurs when plants are concentrates are fed to the animal, and it ends up in the rumen. And during a process, as I said, of feed digestion, the bacteria and other microbes in the rumen digest the feed. And this process results in a production of hydrogen ions and carbon dioxide. If the hydrogen ions are left there, this could result in acidosis, or the rumen becoming too acidic. As a result, methanogens come in and use that, mop up that hydrogen essentially to produce methane. And this process has evolved over time. And this is it's a very essential process in the rumen of animals. However, this comes at a major cost to the environment. This is the essential equation. The CO2 generated by the anaerobic environment in the rumen mops up the hydrogen that's produced during feed digestion, and that produces methane, which is CH4 plus water. This process also serves as a major inefficiency to the animal, as energy is required to produce this methane, and it results in 2 to 12% of loss of feed energy to the animal that could otherwise be used for productive processes. So the saying goes, if you can't manage it if you can't measure it. So we are making great strides here in Ireland in measuring methane in different ways. So the gold standard for measuring methane emissions is the respiration chamber. So these are mainly in AFBI in Northern Ireland. We don't really have any functioning respiration chambers in the Republic. These respiration chambers are essentially a large, big box that the animal can actually walk into, and it stays in there and feeds it within the respiration chamber. And it's gives a very accurate measurement of methane emissions, carbon dioxide, etc. And this is the gold standard for measuring methane emissions. However, if we're doing experiments, we need to have large numbers of animals, and we need them both indoors and outdoors. And as a result, we can use the SF6 tracer method, and this has been used in the past in Teagasc Grange and Teagasc Moor Park to measure methane on pasture-based systems. Also, which is quite popular now, and we use them ourselves in Grange and also in Moor Park, and Ben and Donagh will speak about those in their other presentations to come. And this is the green feed system. And this allows the animal to come to feed at the green feed, and methane is measured during that time. And usually we test animals over a period of 3 weeks for an accurate methane measurement result. So the big question is, how are we going to reduce methane emissions from Irish agriculture? The first is the low-hanging fruit, and this is having improved management practices on your farm. The Teagasc MAC, or the Teagasc Marginal Abatement Cost Curve, which has been put together by Professor Gary Lanigan and Dr. Trevor Donlan from Teagasc with the help of the members of the Greenhouse Gas Working Group. This really quantifies the level of mitigation or methane reduction by a range of improvements or adoption of changes in farm management practices. It also analyzes the costs and the benefits associated with these practices. They found that improved farm management practices can result in methane abatement estimated to be about 1.85 megatons of CO2 equivalents, and this is how methane is measured in our inventories per year. These are cost negative strategies. So these will not cost anything. These will actually enhance your profitability on your farm. And these result in a 10% reduction in total greenhouse gas emissions. So what are these practices? These include extending the length of grazing, increasing your dairy cow genetic merit via the economic breeding index, and Donagh will talk more about this breeding later. Optimizing the age of first calving, increasing your daily live weight gain, optimizing calving and lambing rates so that they can calf and lamb every year, and lowering the age at which the animal is slaughtered so that the animal is not existing on the farm for too long. So really, essentially, all these practices result in enhancing efficiency and productivity on your farm. And of course, as I said, as methane is produced from manures and slurries, we need improved waste management. However, we need to go a step further as we need greater reduction in methane emissions to meet the challenges. Donagh is going to talk later about breeding strategies to reduce methane emissions. I'm going to talk now about one of the projects that I'm leading, Methane, on the development and validation of novel technologies, mainly feed additives and additives for manures, to reduce methane emissions from Irish pasture-based beef, dairy, lamb, and lamb production systems. So within the Methane project, we set out to assess feed additives, as we hear a lot about different claims that are made about feed additives. We set out to look at these feed additives, assess them, examine their efficacy to mitigate methane emissions, while at the same time monitoring their effects on animal productivity in cattle and sheep, and ensuring they have no negative effects on productivity. Some of the feed additives we're going to look at, and we are currently looking at, is 3-NOP, which we hear a lot about, which is commercially available as Bovar. Now, it's not commercially available yet as it's going through the registration process. Seaweeds, oils, halides, which are short-lived oxidative species, extracts like yucca and olive feed, and a range of oils. Ben will talk more about the issues at grazing, but the issue with feed additives is the feed additive needs to be with the feed at all times for it to be effective, particularly in a mature animal. This is often not the case or not possible in grazing systems. And they were faced with a challenge of having to have a feed additive that is there at all times in the rumen of the animal while it is grazing in pasture. Therefore, we will need within Methane to develop encapsulation of the effective feed additives so they can be used in pasture-based systems. Also, it's essential to ensure that the nutritional and the toxicological composition of the meat and milk products from animals fed the feed additives are actually, there's no toxic effects, to confirm consumer safety. And this really, we need to make sure there are no residues from the feed additives in meat or milk products. Also, they need to be sufficiently cheap to be delivered on farms across the country. So the farm level cost effectiveness of any effective feed additives will be evaluated using the National Farm Survey. We've already started this project, and particularly task 1 of the project involves the in vitro or the laboratory-based analysis of these feed additives. This is through the rumen simulation technique, or what we call the RUSI-Tech, in the laboratory at Teagasc Grange. We've looked at a range of feed additives, including the plant and oil extracts, olive byproducts, the halides, which are short-lived reactive oxygen species, which are developed by the company Glassport Bio, which is one of our industry partners, and NUI Galway. We've looked at all of these in a range of concentrations, and also looked at seaweeds in collaboration with the Project Sea Solutions, which is led by my colleague Maria Hayes. We've looked at brown, red, and green seaweeds, and indeed their seaweed extracts. And we've measured methane emissions and the digestibility. So for this rumen simulation technique, we actually use rumen fluid from animals at Grange in the system. We all have heard a lot about the feed additive 3-NOP. This is not commercially available, but there's a lot of work ongoing by the company DSM in advertising its effects. There's been a large number of trials worldwide, and we hear a lot about it in the media. So what is 3-NOP? It's quite a simple structure, as you can see here on the screen, and it's a synthetic non-toxic compound, and the chemical name is 3-nitrooxypropanol, and it's been called a really promising methane inhibitor. It has resulted in consistent methane yield decreases of about 30% in many trials around the world, mainly indoors. So, how does it act? It acts by binding to the enzyme, methyl coenzyme M reductase, which is the enzyme found in methanogens, which produces the methane, and it's the enzyme at the last stage of methane production. It has had positive effects. We've seen positive effects in the literature on 3-NOP in vitro, in vivo, in sheep, dairy cows, and beef cattle. Many trials across the world, but these are mainly performed in indoor systems. The production performance has not been compromised by feeding this feed additive. In fact, we often see increases in feed efficiency of about 5%. We were fortunate enough to be collaborating with DSM, and they approached us in Teagasc Grange to carry out one of their trials as part of their EFSA approval process. So we're going to test 3-NOP in a beef cattle trial in Teagasc Grange in the coming weeks. And we're very fortunate because then we will have a study completed that will allow us to help us get 3-NOP into our inventories as a methane mitigation option if it is successful within our systems. There are issues, however. In fact, these feed additive cannot be used yet on pasture-based systems, and we all realize the importance of grazing and the pasture-based systems here in Ireland as part of our production system. However, they're going to hopefully develop a slow-release prototype to overcome that problem, and that has been currently being developed by DSM and trialled in New Zealand. Also, there are issues with consumer acceptability in that you're essentially feeding a chemical while it is non-toxic and breaks down on the rumen. There is a consumer acceptability issue here. So as part of Methabate, we're carrying out a large number of trials here in Ireland. And the first one, which is ongoing, is in sheep. And this commenced in May and it's being carried out currently in Teagasc Research. We're testing a number of feed additives, including agalin, mutral, oils, the halides, seaweeds, and seaweed extract in sheep. And you can see here we're using the portable accumulation chambers to measure methane within that trial. So we'll hopefully have data from that in September. We hope to carry out a beef trial in early 2022, where we're going to feed ad lib grass silage and concentrates, and the treatments will include the control, 3-NOP, plus the most promising feed additives from the sheep study. And later in dairy and Teagasc Moor Park, we're going to carry out another trial looking at slow release NOP when it becomes available, and the most promising feed additives from the sheep study. And these will be on grey swards, grass and clover. Another area of research I'm involved in is in early life rumen microbiome studies. So in early life, this may present an opportunity for us to manipulate the microbes in the rumen of the animal. Because from birth onwards, the microbes that exist in the rumen of the animal, they're very sparse. At birth, it's essentially sterile. So it's during the birthing process and the environment around the calf when it's born that really serves as the inoculum and the first microbes that exist in the rumen that colonize it and help it to become established. And basically, this happens from birth onwards, and really it becomes more established as it approaches weaning, and then it cannot be changed very easily as it becomes a mature animal. So we hypothesized that we could actually give the animal a nutritional intervention in early life, and that the effects of this intervention, be it a methane mitigation feed additive, and that this would have long-term persistence effects later into the life of the animal. However, we did not know that optimum time frame for the manipulation of the rumen in early life. Therefore, we carried out a study in Teagasc Grange. One of my former PhD students, Owen O'Hara, who's currently working in Canada. And Owen and David did a lot of work in generating a very nice animal model where we were able to investigate the temporal microbial dynamics of the rumen of beef calves reared on 2 different farms. So in this study, we were able to investigate how the rumen develops over time, and also the role of environment as they were on 2 different farms in early life. And we found that the first month of life presents a time frame during which the rumen microbes becomes established, which is a very good finding, because this really tells us that if we have an intervention strategy, it will need to be fed in the very early stages of life. And that this has lasting effects on the rumen functionality, including methanogenesis, which can extend into later life. INRA took this a step further, and Sarah Meal and colleagues in INRA carried out a study for early life in dairy calves, where they gave an oral administration of 3-NOP daily from birth into 14 weeks of life. They found that there was reduction in methane emissions, which persisted to 12 months of age. So even though the feed additive was not fed after 14 weeks, the effects of the feed additive stayed with these animals compared to the controls right through to the first year of life, which was really an amazing finding. And this resulted in a cumulative reduction in around 150 kilograms of CO2 equivalents per head of these calves during the first year of life only. Now, we hope to do this study or a similar study in Teagasc Grange, and to add to it by adding feed additives later in life to these animals that were given the supplementation in early life, to see if we could get additive effects or greater reductions in methane. So the take-home messages from my presentation this morning include: Methane is a potent agricultural greenhouse gas. However, we are working hard in the research community to try to develop and analyse a number of feed additives, and these are being assessed under a systematic approach for methane mitigation potential. Also, we're looking at early life intervention as another strategy to try and reduce methane emissions by early life supplementation without potentially the need to have to feed it in later life. And this would be particularly useful when you consider the pasture-based systems and the problems we have with trying to develop slow release options. Thank you for your attention, and I welcome any questions at the end of this presentation.
Thank you, Sinead, for a very clear presentation and for nice, I suppose, a nice overview of the importance of methane, how it's generated, and obviously would say its potential contribution to global warming. So our next speaker this morning is Dr. Ben Lahart from Teagasc Research, and the title of Ben's presentation is Methane Emissions in Grazing Dairy Systems. Again, a very important, I suppose, topic, as Sinead already mentioned, in that most of the international work is based on animals in confined systems, and we really need to capture, would say, the effect of our management systems and specific, interventions on emissions from grazing animals. So over to you, Ben.
My name is Ben Lahart. So I'm a postdoctoral researcher working with Vista Milk. I'm based in Teagasc Research. So I'm looking into methane emissions in grazing dairy systems. So, our dairy industry is a very, very efficient producer of milk relative to greenhouse gas emissions and methane. I suppose going forward, we need to, come up with strategies of reducing methane. We need to measure that grass. We need to quantify what our cows are doing. We need to understand what's driving these methane emissions, and ultimately need to mitigate it. So previously, we would have found it difficult to measure methane in grazing systems. This is specifically due to the labour-intensive nature of the measurement techniques. Sinead showed the SS6 technique there previously. So, the backpacks on, they were very good, but they only allowed for short measurement periods of a week at a time. So you can only measure methane, 3 or 4 times over a year. So we have the green feeds here in Moore Park for the last 2 years. So we have 4 of these, and these allow for a routine measurement of methane over the course of the grazing season. So cow goes in there, you can see his head, it puts his head in the hood, and air is sampled from his breath, goes into an extractor fan, and finally ends up being measured by an infrared sensor to estimate the amount of methane produced by that cow. So we're going to use these green feeds to look into a number of different areas and how they affect methane. So we're going to look at sward factors, so how the sward changes, in its physiological state over the course of the year. Going from the leafy stage in the spring up to the stemmy reproductive stage in the summer, how this drives methane emissions, comparing perennial ryegrass to white clover, and also potentially looking at multi-species swards. We'll also be looking at animal factors and how these affect methane. So, the size of cows, how much milk they're producing, the age of them, how these all drive methane emissions, and how they also affect efficiency, so methane relative to the amount of milk solids produced. We also developing prediction models from our measurements. So we'll be looking into actually predicting methane in grazing systems, and also comparing, what the current methods of calculating methane are, and comparing these to what we're actually measuring, which is very important. And finally, we'll be looking at feed additives and how feed additives affect methane emissions. There's been a lot of research within indoor systems, looking into potential feed additives, but there's been very, very little done at pasture. So it's very important that we do this and do this research. So this is the methane measurement profile from 2021 this year. So this is from one of our farms. So we started training cows up onto the machine from calving in early February, and we've been outside nearly every week since, with the exception of 1 or 2 weeks in March. And over this period, we have about 26,000 individual methane records from 75 cows. So, it allows us to paint a picture of what's going on, across lactation, as the animals change, their physiological states go throughout the year, and as the grass changes, how these are all affecting methane and driving methane and their changes throughout the year. So this is the diurnal pattern of methane production measured using the machines. So as you can see, methane is quite variable throughout the day. So this is each individual hour over the day measured using the green feed. So as you can see, it's lowest at about 6 o'clock in the morning methane, and its highest, it peaked around 5 or 6 o'clock in the evening. So there is variation there for methane emissions across the day. So it's very, very important that we get as much measurements over this profile to actually quantify how much methane individual cows are producing, you know. So what we found is if we use 2 weeks, a 2-week period to get per cow, you know, so that's about 20 records, we get as much of this as possible. And we get repeatability values, which is essentially how, you know, how good the measurement is and, you know, how much it changes to the next measurement. So repeatability of 1 is ideal, but, you know, the values we're getting at 0.69 are very good and are comparable to the repeatability values of milk yield. So it just shows that the data we're getting at grass is of a good quality. We look then into the methane profile of one of our farms here. So this is from early March up till late June. So you can see methane is changing quite a bit. In March, they were inside for a week or 2 there. As you can see, it's a spike there. There was silage in the diet. But then going into April, as the cows went into their second grazing rotation and into the third grazing rotation in May, you can see that methane kind of dropped a bit. It went down to 255, 260 grams on average. And it stayed quite low for those 2 months. So, you know, as the grass appeared to be, you know, quite leafy, methane emissions were reduced as a result. And they appeared to climb then going into June. So as the grass became stemmier, it lost a bit of its feed value, the methane emissions of the cows began to increase. And this is, we've seen the values, you know, being very, very high last year as well, at about 350 grams. So, but they are lower in spring. So how does this compare, the measurements we're seeing, how does this compare to our current methods of calculating methane of dairy cows? So at present in Ireland, we use an emission factor to calculate the amount of methane produced by individual dairy cows. And this is used in our national inventory to quantify the amount of methane produced by the average dairy cow in Ireland. And it works based off an emission factor, which is derived from a number of different study populations, which were fed high forage diets. So the intake of the cow is predicted using its maintenance requirements for milk production and live weight, as well as live weight change. And this intake is extrapolated to the gross energy value of the diet to get its total gross energy intake. And then the emission factor, which is the assumed amount of energy lost to methane, which is 0.65, so 6.5%. This is basically used to calculate the amount of methane produced per cow. But how does this compare to the values we're seeing actually being measured per cow? So when we go back to the graph there and do it out by month, we can see there in March, April, and May, there's quite a difference between what the cows will have been calculated to do based on the calculation to what we were actually measuring. So we're seeing a 67 gram difference in March in that the calculation was overestimated by 67 grams. In April, it was a 105 gram difference. And in May, it was a 97 gram difference. It began to converge in June. So the measurement calculation and the measurement began to slightly align more, but definitely in that early lactation period, it appears that we may be overestimating the amount of methane our cows are producing. This is during the peak milk months, so the calculation is, I suppose, biased towards increased milk yield. And what's driving this milk yield is very, very good quality grass, very good quality grazed grass, which appears to be actually driving down methane emissions. So when we look at Farm 2, we've seen a similar scenario where that methane was reduced from the April to May period. And we did the calculation again. There were differences in that the calculation was overestimating methane on average across March, April, and May, and the calculation measurement were close in June. So it just suggests that the level in which we're calculating and the difference between the measurement is likely to be overestimated in the early lactation period. We need to follow this over the course of the year to see how it averages out, how the difference looks on average across the year. Potentially, we may need to go into seasonal specific calculation periods in that we calculate methane specifically in the spring, summer, and autumn potentially. But this will become clearer as we get more data on this over the coming year and into the next few years as well. So then looking into animal traits influencing methane emissions. So we have this is from 2020 data, and we just looked at the different animal traits that are driving methane emissions and efficiency. So first graph is lactation number. So older animals produce more methane. It's not shown here, but they produce more methane than younger animals. But in terms of their efficiency, they produce less methane per kilo of milk solids output. This is essentially because they're diluting their methane emissions and they're more productive and more efficient as a result. Lighter animals as well will produce less methane per kilo of milk solids, so they're more efficient producers of milk solids relative to methane, and animals produce more milk solids will produce less methane per kilo of milk solids. Again, this is similar to the lactation story in that they're diluting their methane emissions because they're more productive. We take it a step further and look at the EBI, the Economic Breeding Index, and what we're breeding our dairy cows in Ireland for at present, and look into the individual traits that we're selecting for. You can see we're already selecting for increased efficiency through the milk solid subindex, increased efficiency through the fertility subindex, in that we're breeding older cows, and increased efficiency through the maintenance subindex, in that we're breeding slightly lighter cows. We're breeding for increased efficiency through the EBI. So to take this a step further, we recently published a study in the Journal of Dairy Science evaluating the total greenhouse gas emissions effect of selection on the EBI. So we used the next generation herd in Teagasc Moorpark here, which is essentially a herd set up to evaluate what genetic selection for the EBI would do. So it has two strains of Holstein Friesian. One is an extremely high EBI group, €180, and one has an EBI group of €80. So it's €100 difference between the two groups, and the elite group is about 10 years of genetic gain ahead of the national average group. So in addition to methane, we also looked at nitrous oxide and carbon dioxide emissions as well to get a total greenhouse gas emissions profile of the two groups. As you can see there was no absolute difference in greenhouse gas emissions between the two groups, but the elites were more productive. They were producing 10% more fat and protein corrected milk. This is due to their increased age structure and their increased genetic potential for milk solids output. And when you combine it with their greenhouse gas emissions, there was a 10% reduction in greenhouse gas emissions per kilo of fat and protein corrected milk compared to the national average group. This is essentially because the elites were diluting the amount of greenhouse gas emissions they're producing. So for every €10 increase in EBI between the elite and national average, we observed a 1% reduction in greenhouse gas emissions per kilo of fat and protein corrected milk. So this is the annual rate of genetic gain for the EBI at present in Ireland. So for each year, farmers continue to select the EBI to want to reduce their greenhouse gas emissions intensity by 1%. But it's important to highlight a present EBI won't reduce greenhouse gas emissions on an area basis. So potentially need to select for traits such as methane and EBI. Donagh is going to cover this in the next presentation. So then finally to touch on feed additives. So feed additives have been widely researched within indoor settings. Loads and loads of studies published internationally on the potential of feed additives within indoor settings. And there's been large reductions observed, in red seaweed, feeding animals red seaweed have seen reductions of 80% in enteric methane emissions. Sinead mentioned Trinop there, and Trinop, shown values of 20 to 30% reduction. But it's important to note that these feed additives have been fed, in total mixed ration diets, which essentially allows the additive to be mixed continuously through the feed. So for every given mouthful of feed the animal is consuming, it's going into the rumen, and it's having a mitigating effect on methane emissions. At grazing, we're more constrained in that, in a typical Irish grazing dairy system, we can only supplement cows with the likes of feed additive and concentrate twice daily at evening and morning milking. So it's important that we do research into the potential of feed additives in these scenarios. So we conducted two experiments in 2020 evaluating two different feed additives and how they influence methane emissions in grazing systems. So in the first study, we looked at a group of phytochemicals, and in the second study, we looked at a blend of seaweeds. So the seaweeds were red, green, and brown seaweeds. And we looked at their effect on methane emissions and milk production. And within the two independent studies, the additive was fed with a kilo of concentrate. So there's 1 gram of phytochemicals within the first study per kilo of concentrate, and there were 70 grams of seaweed per kilo of concentrate in the seaweed study. And the two of these were compared to control. So the concentrate were fed at evening and morning milking a kilo each feed to basically simulate what would occur in commercial practice. What we observed was there was no absolute reduction in methane emissions with the phytochemicals. There was a tendency for increased milk solids output. In the seaweed study then, there was no significant effect detected for methane emissions, and there was no significant effect detected for milk solids output either. I suppose One of the reasons potentially why we've seen no effect was going back again to the fact we were only feeding it once to twice daily. And any potential effect was disappeared, after an hour in the room, or it didn't even have time to have an effect. So it's important that we develop slow release formulations for grazing systems, as Sinead spoke of, to allow the additive more time to have an effect within the rumen and change that rumen environment to reduce methane emissions. So the conclusion then is sward characteristics and methane need more research. So the values we're seeing, that reduced methane output we're seeing in early lactation in the spring period, we need to do more research to understand what's driving that. We need to do more research into sward characteristics that will reduce methane across the whole grazing season. There is significant potential there to improve efficiency and reduce total methane emissions through the sward. We're going to be doing more research on that. At large, EVI is breeding more efficient animals. It won't reduce greenhouse gas emissions on an area basis, but will improve efficiency. And finally, additives then, we need to focus on additives that are suitable for grazing systems and to develop such additives, to be used in grazing systems. So that's it there. And if anyone has any questions, you can ask them at the end. Thanks very much.
Thanks very much, Ben, for a very clear presentation, comprehensive presentation, and for highlighting, I suppose, many nuances, would say, that you can be presented with, would say, in terms of, trying to capture, would say, emissions from grazing-based animals, which is absolutely critical, would say, for inventory development. And it just highlights, I suppose, the importance of homegrown research, would say, and the necessity to do our own work that's applicable to our own systems. So next up is Professor Donagh Berry, and Donagh obviously will be known to many of our attendants this morning, and he's going to speak about his work examining the potential for breeding strategies to reduce methane emissions from ruminants. And again, it's very complementary, would say, of the previous two presentations, which focus more maybe on the systems and also a nutritional intervention approach. So over to you, Donagh.
Okay, thanks, David. So if we think about breeding, breeding is a really powerful tool, and we have great examples of this for the past few decades, even for the past few centuries. I'm just going to give you a quick overview from what we have achieved from breeding programs in different species. This lady here, she's the Guinness Book of Records holder for the amount of milk produced. And look at this, look, 32 tons of milk, over a ton of fat and almost a ton of protein produced over a 365-day lactation. That's the equivalent actually of the calorific value of around 26 steer carcasses per year. Now, obviously, that's not all due to genetics. Management has a huge role, and nutrition has a huge role to play in this, but the fundamental genetic basis of this cow was crucial. If we look at some of the studies that have actually tried to partition out nutrition from genetics, this is a great example often used by geneticists. Poultry, obviously, from 1957 line on the top and 2001 line on the bottom. And those numbers, 43, 57, 71, 85, are days of age of those two different strains. Fed exactly the same, managed exactly the same. There's no need for me to put words to that. I think the picture itself demonstrates clearly the power of genetics in achieving performance gains. Another example from the chicken industry, 1950 line on the left-hand side, 68 days of age, really scrawny, versus this huge plump animal at 2008 strain at 47 days, so 21 days younger. Now, that's not all to say that breeding is absolutely brilliant. These animals on the right had issues, particularly cardiovascular issues, and issues with the strength of their feet, actually the ability of their legs to hold up those different breast weights was difficulties. And then finally, moving on, beef, the size of our animals now, the muscularity, most of this would actually be due to genetic influences. So really what we're talking about in this talk is the ability to harness that potential of breeding to get this cumulative and permanent benefit over generations. Before I dig into talking directly about methane emissions, just to quickly go through some of the fundamentals of breeding. And one of the fundamental components of breeding is that any performance trait that we measure follows this type of a distribution. So let's just take human height, for example. Many of us are there in the middle, right? But as soon as the pubs open, you look around the pubs, you'll see that there's always a few small people and there's always a few tall people. Incidentally, if you are in the pub and there's a pub quiz, the average human height for males is 175 centimeters. And the top 10% of men in the world, their average height is 187 centimeters. So a big difference between the extremes. And that's humans. If we look at sheep or if we look at cattle, we can look at their carcasses. So there's going to be carcasses that are in around the middle. That's where most of them are, around the average. There's going to be really small carcasses. And to the right-hand side, there's going to be really big carcasses. Similarly, if we look at dairy cows or sheep, milk yield is exactly the same thing. Most cows produce the average. That's the definition of the average. And then, of course, we have cows that produce less milk and cows that produce more milk. Now, why I'm going through all this with you is that if you think about all those traits, human height, milk production, carcass weight, we can actually measure all of those things. So it's easy to identify which animals are on the right-hand side of the distribution versus the left-hand side of the distribution. And as geneticists, what we try to do is dig into that data to identify, well, this animal that's milking a lot, is it milking a lot because it's got good genes or because it's fed to milk a lot? But to do that, we need a lot of data. And Sinead would have talked about that at the start, and I'm going to come back to that at the very, very end. So when we talk about breeding for methane emissions, I would separate it into a dual approach. So there's the indirect approach, what I would call that the low-hanging fruit. And then on the right-hand side, there's the more direct, more going towards methane emissions specifically. And we've heard from the lads that that's a difficult thing to do, to measure these traits on sufficient animals to enable a breeding program to be useful. So what I'm going to do for the rest of the talk is take you through a journey through both of those, starting off with the indirect approach. This is a very, very simple illustration of methane output per day of age as an animal ages. This is an example of a steer, age on the bottom. It could, of course, be a lamb or it could be any growing animal. So an animal produces methane every day, and that culminates over time. So as the animal gets older, it's going to produce more and more methane. But what if we were to shave off, say, the last month of life of a particular growing animal, be it a lamb or be it a bull, steer or heifer? There is a potential there then to reduce the methane associated with that lifetime of that particular animal. In fact, if you were to the last month of life, based on some of the data that we already have in beef cattle, is the equivalent of just over 200 tons of carbon. So if we were to shave off potentially 5 months, now I don't think this is achievable in the near future, but if we were to shave off 5 months of the age at slaughter of an animal, we could actually achieve the mitigation target, the minimum mitigation target that has been set down currently for agriculture. If we look at this from a genetic perspective, actually we were the first in the world to show that this is under genetic influence in cattle, and it's around 30% genetic. So there's a massive potential here to indirectly improve the methane emissions of an animal via shortening of its age and therefore its cumulative methane output. If we look at mature animals then, again, be it sheep or cattle, on the vertical axis here, I just have a variable called the amount of calorie output. That's just to account for milk versus meat. So the amount of calorie output per kilogram of carbon dioxide. As most of us would know, in what we call a nulliparous animal, or an animal that has never had an offspring, so a ewe lamb, a hogget, or a heifer, dairy or beef, doesn't make a difference. All it's really doing is eating and producing methane. So it's bringing it into the red. It's not producing any product that you can actually sell if you're keeping that animal. Then once the animal gives birth, be it lambs or calves, it will start producing calories, be it in milk output or the growth of a young animal. So it's recouping a lot of the actual cost, the initial cost or investment in methane in that animal when it wasn't producing anything. And that's the black there. And there's an equilibrium point then, wherever that may be set out at, that once you get above that, then the amount of calories it's producing is over and above its carbon dioxide contribution over that time period. So if you can get that animal to produce more and more for longer and longer, you're diluting that red part at the start of the animal's life over a long time span or a greater amount of calories. And that's due to increased longevity, but it's due to also increased output per lactation. And that is really what we're trying to do in breeding. What I'm demonstrating you to here is the 6 different breeding objectives that we have in Ireland. So we have one for dairy, which the lads Sinead and Ben already talked about. We call it the Economic Breeding Index, or the EBI. Below that is our Dairy Beef Index. That's to select beef bulls for use in dairy cows. Then we have a replacement and a terminal index, both for beef and also for sheep. And this is a different emphasis on the traits within those different breeding objectives. So there's 3 points I want you to take from here. If we just firstly look at the mature animals, look where all the emphasis is on those animals. It's on reproduction, greater longevity, getting the animal to live for longer and longer. But it's not just that. If you look at the orange highlight there, that's on calorie output. So it's milk output or meat output. So it's getting the animal to produce for longer and longer, but more and more per lactation. So its lifetime calorie output is greater. That's the first point. The second point, and Margaret Good actually made a comment on this, is the implications of a lot of our mitigation strategies on animal welfare or other traits, is we don't select on any individual point or any individual trait. It's within a holistic breeding objective. So if we were to select, for example, for methane, it wouldn't be exclusively for methane. It'll ensure that there's no implications or no repercussions on animal longevity or output or beef carcass traits, etc. And then the third point that I want you to take from this is if you actually look at those 8 sub-indexes or those components of those indexes, there's no mention of environment, no explicit mention of environment. Yet I'm going to show you in the next few slides is through indirect selection. So remember, this is this low-hanging fruit. We are actually achieving gains in performance. And Ben actually alluded to this in the middle of his presentation as well from a dairy perspective. But I'm going to cover dairy, beef, and sheep. So let's start off with dairy, a bit of a reiteration of what Ben talked about. Ben was looking at one particular herd, 2 groups of animals, what he called the national average and the elite. But I'm looking at the national population here. And this is the national dairy cow population in Ireland from the year 2000 to the year 2020. This is actually when we introduced the Economic Breeding Index. It's introduced 20 years this particular year. And look at the gains that we have achieved. If you look at the blue line, that's how the EBI has increased over that time period. So we can see today that our animals are around 160 euros higher EBI than what they were 20 years ago. Economically, that equates to cows today being 320 euros more profitable than what they were 20 years ago for exactly the same management. This is exclusively due to genetics. And we can see that was achieved by this indirect approach of improved fertility, which is the green line, so getting cows to live for longer and longer, and produce more per lactation. Ben mentioned this point. If we actually take Ben's data and extrapolate it out to the national population, this improvement has equated to a 14% improvement in the carbon footprint per kilogram of milk solids. Also, actually, and this is also Ben's work, it has also improved the nitrogen use efficiency of those animals, but I haven't discussed that here today. If we look at it from beef, again, on the bottom axis here, I'm showing you these breeding indexes. I just picked the terminal index here and on methane output on the right-hand side. This is some of the data that Claude Ryan is collecting from the Irish Cattle Breeding Federation in Tully. And the dots here, those dots represent an individual animal. The colors represent different genders. There's steers and heifers represented here. So if we just look at that green arrow, that animal there has a terminal index of around 60, 70, and is producing around 125 grams of methane per day. So if we follow that trend, you can see the line is going down to the bottom right. That what that is saying is that if you increase the terminal index of your animals, you will reduce the daily methane emissions. But again, this double whammy effect is you'll actually also be more profitable. So if we were to compare, and I just went on to the AI bulls available through the Irish Cattle Breeding Federation, compared the top 10 bulls available versus those from 90 to 100. So the bottom 100 bulls. The difference in carbon dioxide output per day of those animals is over half a kilo. So that's a 10% reduction. But the farmer would also, on average, make 63 euros profit per animal more by using that strategy. So again, it's more profit for the farmer, but also reduced environmental footprint. Sheep, exactly the same story. Some of the work that Noreen McHugh is doing up in Athenry, we have a star system. We have it in beef as well, but just looking at it in sheep, where it's just like the hotels. We have a 5-star hotel there, a 5-star sheep. That's your top 20% of sheep. they on average, according to Noreen, produce just under 8 grams of methane per day. While we have the 1-star sheep, the bottom 20% genetically all manage the same, so the bottom 20% genetically produce almost 8.5 grams of methane per day. That's around a 7% difference in methane output between the two groups. But again, the 5-star animals were more profitable. We expect them to be around €12 more profitable per ewe. So again, this double benefit. So that was the indirect approach, the low-hanging fruit. If you look up at the top right there, now we're into the high-hanging fruit, the stuff that is at the very, very top. This is the directly selecting for methane. This is just simulation data based on some of the parameters that you'd see internationally. On the bottom, it's not really that important, but it's calorific output, so it's milk yield or its growth rate doesn't really make a difference. And on the vertical axis, it's methane production per day. What we would expect and what we see internationally, and again, some of the stuff that Ben would be showing is that increased milk output gives you, on average, more methane production because the animal is eating more. The question that we're posing from a breeding perspective is that if you just take an animal or take animals that are growing at 20 or a milk yield of 20 kilos per day, right? You can see some animals there are producing 300 grams of methane, while there's other animals that are producing exactly the same milk output. They're actually almost double the amount of methane being produced. So our question is, well, what is this variability? As I said, I just simulated this, but is it double? Is it half? Is it 300 grams? Is it 200 grams? And importantly then also is how much of that is actually genetic. And it's then, and only then, can we decide whether it's worth having a breeding program to directly target methane output. Sinead talked about the measurement of methane. This is just my last slide. As you can see here, the ones on the left, these are our green feed systems for measuring methane outdoors. In the middle, we have the same system, but it's for measuring methane indoors. And we have what we call these portable accumulation chambers or pack chambers on the right-hand side for measuring sheep. So currently in Ireland, we've methane measures of around 150 dairy cows, around 770 beef animals, and 950 sheep. Now, if you want to select on a trait, and in this situation, we're talking about methane, there's 3 things that must fulfill before we would consider breeding for it. Firstly, it must be important. Secondly, it must have genetic variability there. So there must be genetic differences. That scatter plot I showed you, how much of that is due to genetics? We call that thing heritability. And then thirdly, and I would argue more importantly from a methane perspective, is we must have sufficient data available to be able to disentangle what are the good genetic animals versus the bad genetic animals. So just to conclude, I think when we talk about sustainability, a lot of the narrative revolves around environmental sustainability. But for me, key importance is social sustainability. So in other words, embarking on these strategies must actually return an investment. And that's the real beauty about breeding, which I've demonstrated to you, is a lot of the tools that we're using, albeit indirect approaches, are reducing methane output, but they're also improving profitability. We look at livestock, and again, it's all over the media about the contribution of agriculture to environment. Looking at the data that we've shown you here today, I think I'd look at that and say there's a massive opportunity here to actually reduce the environmental footprint. The benefit about breeding over other nutritional or management approaches is what we call as cumulative and permanent, in that you build on it year on year. And traditionally, we would have looked at breeding as being very slow. But we've quite clearly shown that for the past few years, it's not slow. And with sufficient data underpinning it, it can actually be quite rapid. So we can make rapid genetic gains. And just to finish off, and I guess it's kind of tying all the presentations together, I don't believe there is any one silver bullet, and it has to really be a co-evolution of both animal level contributors, like what we heard about, what Sinead was talking about from a feed additives perspective, farm, like Ben stuff, the real good stuff on the grass, and also animal breeding. So thank you very much.
Thanks very much, Donagh, for a very comprehensive, and complimentary, I suppose, presentation. It's, I suppose, very good to see that, breeding, essentially complements obviously the nutritional strategies that that you mentioned there, but equally would say that many of the traits that we are selecting for would say there's no obvious, antagonisms with the methane trait. Essentially, more productive, more efficient animals, would generally have a lower methane intensity. So I'll open the floor now to questions. And again, it's great to see the number of participants. You know, we're up around 150 participants there tuning into this session, and equally the number of questions. So again, and I'd encourage you, would say maybe over the next 10 minutes or so to, you know, to upload your questions to the Q&A session. So I'll kick off maybe just in terms of maybe our chronological order this morning, a question, you know, it's probably relevant to everybody, but I'd start directed to Sinead maybe firstly, from Fern. Sinead, are the results of, you know, the trials that you mentioned there, are they publicly available to view? You know, and obviously would say those that are conducted in the future, and you mentioned would say certain maybe proprietary products that will be used, will those results be available, would say, to the public?
Yeah, thank you for that question. Absolutely. So once the trials are finished and we analyze the data, our aim is really to get this data published, and that's when the data will be viewed. So yeah, all the data from these trials will be made publicly available. They're actually PhD students working on these trials at the moment, and they will need this data for their PhD thesis. So absolutely, once it's analysed and published.
Okay. There's a couple of questions, would say, looking at the potential of grass quality and also variety. I might maybe focus that question to Ben. You know, a couple of people asked Ben, would say, whether there's work looking at the effect of, I suppose, grass quality, grazing management, and also variety, you know, of ryegrass. And I suppose essentially would say, you know, we hear a lot about mixed species swards as well, you know, and that has come in a couple of the questions. You might just comment on that, would say, in terms of what you might, you know, your knowledge from the literature, from the work that you've done.
So like just to touch on the values we've seen there in the spring, you know, that was very, very good quality grass. No, we don't know the exact reasons why it was lower, but likely due to increased feed value, you know, increased digestibility, reducing those methane values. You know, I suppose there's also work done internationally looking at levels of nitrate, feeding nitrate to cows, and that reduces methane emissions. And spring grass potentially has high levels of nitrate, which may have a mitigating effect on it as well. Just moving on then to grazing management strategies, I suppose going into lower covers is the best way of reducing methane per kilo of intake, increasing digestibility of the sward by increasing the leaf content, reduce methane per kilo of intake, and increase efficiency, and it'll reduce methane per kilo of milk solids output. There is research being conducted in New Zealand looking at potentially breeding grass for higher lipid content, which will have a higher energy to reduce methane. You know, so this will be interesting work to follow. And I suppose, like, if we can engineer a sward that can reduce methane, potentially that will have a massive effect, because the animals, you know, I was saying the limitations of additives in that they're limiting grazing scenarios that the animals can only consume them twice daily. If we get a sward that can have an effect for every mouthful of grass eaten that could mitigate methane, you know, that'd be massive. Like, so definitely is really important research, and it's important that we follow it to see what's driving it.
Good, thanks, Ben. A question from Margaret for Donagh. Donagh, I know you alluded maybe to some of this already, would say, but, you know, has the potential impact of, I suppose, a reduction in greenhouse gases, you know, an impact for increasing the longevity, would say, in terms of, I suppose, lactation number, would say, for some of the highly productive cows? Has that been investigated, and is it something that you feel would be worth looking at?
Yeah, so like what we're trying to do is, as I said, the dual objective of increasing the amount of milk produced per lactation and the number of lactations for which the animal does that for. Now, that is really back to Ben's point, it is within the confines of a grass-based system. So that cow that produced 32 tonne of milk, that is not a suitable cow for Ireland. And if we bring that cow into Ireland, then we're going to have to bring the feed associated with it, which, when you do this thing called complete life cycle analysis, so what is the cost of actually growing all those alternative crops, importing them into the country, let alone the security, the food security aspect of associated with it, such. I think we need to select for let's call it mediocre at a global level milk production that can be fulfilled from the stuff, the grass that Ben talked about. Like, to me, one of the real take-home messages I got from the whole seminar is, to me, the new feed additive is actually grass. That's what Ben is telling us. It's high quality grazed grass, and the potential there to reduce methane from that is absolutely phenomenal. But it's my last point, the co-evolution has to be there, is we need to have the cow that can ingest a sufficient quantity of that grass to meet its nutritional requirements.
Thanks, Anna. Just maybe a secondary question there, and you alluded to it there both in the presentation, I think in your answer there. Just, I suppose, the concept, and I know you're doing a lot of work down to the years looking at, I suppose, the interaction between genotype and the environment or the production system within which the animal is placed. Do you feel that from a methane perspective or greenhouse gas perspective that, that's something that can, I suppose, add greater complexity, would say, from an Irish perspective there in terms of trying to capture methane emissions for our national inventories?
Yeah, so there is this phenomenon in breeding called genotype by environment interaction, which means that the difference between two animals or two breeds or two genotypes of animals differs depending on the environments. That happens for every single trait. It's the extent of which it happens is how complicated it makes the analysis. So one of them is where the difference is just larger or smaller between two breeds. And we would have seen that in Ben's stuff, actually, even within the 1 year, because the variance changes with the mean. So I suspect, Ben, the difference between your animals is a lot less in April, and it's going to be a lot more in June. Now, do we want to consider that and make breed for animals that are producing different methane in different types of years? I don't think so. The other genotype by environment interaction is where the animals flip over. So the animal is really good in an indoor system, produces a lot of methane, and then it produces the least amount of methane in a grass-based system. I find that hard to see where there'd be sufficient amount of re-ranking, and we don't see this in any trait, where there'd be sufficient amount of re-ranking to justify a breeding program to be separated for each.
There's I suppose, a common theme among some of the questions there in terms of, I suppose, the ability, would say, of us to measure, emissions, ultimately, would say, be able to capture those, to an extent that would be, I suppose, harnessed or exploited within the national inventories, and obviously get credit for that, whether it be sequestration, would say, or direct mitigation policies or opportunities. So maybe I might just direct that at Sinead, would say, in terms of, what's typically required, Sinead, would say, within the context of these type of research products to ultimately get that data, that information captured, would say, and be of national benefit.
Yes, that's a good question. So really, in the past, we would have had very few, say, data points for methane emissions. So really what we need is to have methane emissions data for our production systems. And as Donagh and Ben mentioned, particularly at grass-based systems. So we're starting to gather those now. So rather than having to use methane emissions data from other countries and other production systems, we need to gather our own. So that's actually happening. And Donagh mentioned the kind of numbers of data we're collecting, and that will be very useful for the EPA in putting together the inventories. But in terms of mitigation strategies, in order for that to be as part of our national inventories, the actual mitigation needs to be trialled in Ireland for it to be the national inventories under these production systems. And there would need to be at least a number of publications so that it could be used. Now, we have seen where it was just one publication in certain cases, and that would be then taken on board by the EPA and used in our inventories. But I think really what we will need for that mitigation strategy to be applied in the inventories, it needs to be trialled and the data published beforehand. The other thing to consider as well, for feed additives, they would have to be registered. So for use, and they would have to be gone through the EFSA approval process. And I know there's some comments there about safety and welfare and organics, etc. The additives for them to be used in Europe, all that information in terms of residues and their safety would be carried out beforehand before it could actually be made available as a feed additive.
And I suppose building on that, there's a question in from Victoria, directed at either yourself or Ben, but maybe I'll ask Ben. Whether there's potential, risks, would say, with the use of some of these additives, and maybe the question is targeted more towards the likes of seaweed and others, in terms of feeding those within the context, would say, of a pasture-based system and its potential impact, would say, on the quality or indeed the processability of milk.
Yeah, so that's very important. That's very important to be aware of. If we find an additive that works, it's very important that it's assessed to see how it affects, the milk processability, meat quality, animal health. Like some of the research there, like red seaweed, there's been some studies showing that there's increased levels of bromoform in the milk as a residue, which, isn't ideal. And there's increased levels of iodine in the milk, which would affect its, ability, you need low levels of milk iodine for infant milk formula, so that it affect the export potential of that product. In terms of health characteristics, and there's a study recently published on Red Sea which showed it actually had a negative effect on the rumen wall, the health, the rumen, the papillae in the rumen wall. So it's very important to be aware of this, and if we do find an additive, to assess how it affects the total system, the total system effect in the context of pasture-based systems.
So in the interest of time, I'll just direct maybe a last question then to Donagh. Stuart asked Donagh, I would say there, in terms of, I suppose you highlighted the necessity for large numbers, would say, of, I suppose, methane traits and obviously, or methane measurements, and obviously it's a trait that's not routinely measured at farm level. Is there any proxy traits or what, I suppose, resources or infrastructure would you require, would say, essentially, would say, to be able to, include, would say, methane trait within the national dairy and beef indices?
Yeah, I actually answered that on the chat, but
Okay, sorry.
But if we forget about the proxies, like our dairy and our beef industries in themselves are multi-billion euro industries which are under investigation. How we cannot invest a few million euros on actually measuring true methane on these animals is beyond me. And I think it's something that we need to consider. Yes, the cost has to be incurred by the guys at the top of the breeding pyramid, but it'll be realized by all the guys in the bottom. So if we actually divide the total cost, resource cost, by the amount of liters, billions of liters we produce, or tons of meat or sheep or beef meat that we produce, it's actually very, very small. But to answer the question, infrared spectroscopy of milk, so every cow is milked twice per day. What happens is, like when you go to the doctor, they tend to take a urine sample of you, they look at it, and that gives you a bit of an idea about physiologically or metabolically what's happening inside in your stomach. Same in cows, you're getting access to biological sample twice per day. Obviously, you can only do milk infrared in milking sheep and dairy cows. So there's other things like maybe doing a similar process on the poo of animals, so the beef and the sheep. Ben has done a bit of work on that in his PhD. And also these things like lasers, shining the light on lasers, and based on same technologies, infrared spectroscopy actually, it's the reflectance of the waves as they hit the animal. I don't know, are they any good or any bad, but to me, Stuart, I would say, let's just put in a load of green feeds and start measuring this stuff.
I think, we need to up the ante, I suppose, as you say, would say, in terms of getting a handle on, the variation between animals, but equally would say the variation even within an animal throughout the year has very, very clearly presented there, would say, in his work in terms of both, I suppose, the effect, would say, of pasture, but equally would say, the lactational process as well and its impact on dry matter intake. So I think that's a nice, conclusion, and I'd like to, I suppose, draw this morning's webinar now to a close. As Donagh alluded to there, a lot of the questions, and there have been many, many questions, and again, some common themes amongst the questions as well, which is, it's great to see. The various presenters will be in a position to actually address those, within the chat function after the presentations. And as I said to you, all three presentations will be available very shortly on the Teagasc website. So I'd like to, I suppose, thank sincerely, would say, our three speakers this morning for the very clear and thought-provoking presentations, and particularly to yourselves, would say, the audience. And as I said, there was over 150 people, would say, on this webinar this morning. Thank you for your interaction, for the clarity of your questions, and I suppose before we finish, I'd like to also draw your attention to the next webinar in this series, would say, and again, that would be out, the content of it, would say, the speakers will be outlined in the slide in a moment's time. So with that, I thank you again for your attention and wish you a good day. Thank you.